It's my pleasure to introduce our next round of speakers. And our fourth and next speaker is John Scott from the University of Washington. And he's going to give a talk entitled Exploring and Exploiting the Spatial Constraints of Local Signaling. Welcome, John.
Thank you. And there we go. So first of all, thank you very much to the organizers for this invitation to come and talk to you today. It's just really wonderful to be able to hear about all the fantastic local science that's going on and the kind of camaraderie that can be developed from that. So to begin with, this is an artist's impression of the inside of a cell. And this is work that was done by David Goodsall at Scripps Clinic. And it's to scale, it's called mesoscale. It's about a 2 million-fold magnification from an electron micrograph. And these are all the proteins that we've been hearing about. And the important thing is, if you look at this, there's not a tremendous amount of space for things to move around. But we know that that happens inside a cell. And since my lab is particularly interested in the molecular and cellular aspects of cell signaling, what's interesting is in the context of this sort of tapestry of proteins, you have signals that diffuse from one point to the other. You have covalent modifications. You have mechanotransduction. And the question is, how are these processes coordinated and controlled, and how do they go wrong in disease? And that's really what I'm going to talk about today. And the story I'm going to tell you is really in the context of a family of molecules that we've been working on for a long time called A-kinase anchoring proteins, or Ecaps. And they function principally to target broad specificity protein kinases to specific locations in the cell, particularly the cyclic AMP-dependent protein kinase, or protein kinase A. Now, this is something that I'm pretty sure that all of you, including myself, learned in undergraduate biochemistry, that the cyclic AMP-dependent protein kinase consists of a regulatory subunit dimer and 2 catalytic subunits that are released upon the binding of cyclic AMP and then are available to sort of diffuse through the cell. Now, what I'm going to tell you is that that's probably not the case, because these Ecaps function to focus and insulate cyclic AMP signaling to extremely defined microenvironments within the cell. And that's kind of shown a little bit over here, which is a proximity ligation analysis of an Ecap pKa complex in pink and the Ecap itself in blue. It just happens to be called Ecap1. There are about 50 of these Ecaps in the human genome. Now, the important take-home message from this is that you can see there are many more blue spots than pink spots. So there's many more free sites for anchoring than there are anchored pKa complexes. Now, when you add to this that we know that the binding affinity of this protein-protein interaction is somewhere in the order of sub-picomolar with little or no off rate from in vitro biochemistry, it opens the possibility that all of the kinase is tethered to these Ecaps in proximity to particular substrates at discrete locations within the cell. So that becomes particularly important in terms of the... My computer wants to know where I am at the moment. Okay, I will allow that so I can continue my talk. So what I'm going to tell you is, rather than this model for dissociation, what we believe is that you actually have a solid-state signaling complex. Very, very subtle difference from what's been shown before, but has very profound functional ramifications. And this work really began from cryo-electron or single particle EM analysis of some of the Ecap macromolecular complexes. Now, David showed some much more beautiful images than this earlier, but these are much smaller images of a pentameric Ecap pKa holoenzyme complex. And when you look at this complex in particular, if you look at it to scale, you can begin to put together the molecular jigsaw of these 5 proteins. The Ecap is in gold, the regulatory subunits are in green, and the 2 blue parts are the kinase moieties themselves. Now, the important part here in some ways is not the ordered structure of these different domains. It's the region of intrinsic disorder between the domains, because this creates this element of flexibility. Now that's something that X-ray crystallographers absolutely hate because they can't crystallize it. But when you use single particle EM, you can begin to look at the range of conformations going from one energy state to another that gives you that range of flexibility within this macromolecular complex. And you can begin to see this here, where we've ordered these from the most compact to the most extended. Now, again, when you look at this, the important point is that this is happening within the confines of the cell. And it's happening in a dynamic equilibrium between this movement of these complexes. So you can actually begin to model this by taking real data and creating these movies, these QuickTime movies, that show a range of motion of between 200 to 400 angstroms. Now that's a very important number. It's about the width of two molecules within inside the cell. So if you think about that in terms of how these molecules work, if you have a substrate that's bound to the complex, it would be easy to get multi-site phosphorylation, particularly in the kind of tight conformation. But in the extended conformation, you could have the two different wings of the complex servicing different subcellular environments to increase the propensity for phosphorylation. So again, all of this happens in the sort of molecular congestion of the cell. And if we go back to David Goodsell's image, here is approximately, on the sort of back of the envelope calculation, 200 angstroms. So it's not very far for this system to have to move. And that's why the idea of signaling being enhanced through this intact holoenzyme is very, very important, because it not only allows you to focus the signal at precise locations within the cell, but it allows you to insulate the signal so you can have parallel processing in response to different messengers at the same time inside the cell. So effectively, the cell can multitask. So all of that really sort of flew in the face of this very conventional model of the cyclic AMP signaling cascade, where the idea that the catalytic subunits are released to do their phosphorylation. But the issue that was always complexing or confounding to me was, how did the system refold? And it's inefficient to allow these free enzymes to float around the cell. In fact, as I'll show you at the end of the talk, it might be a pathological mechanism. So to really begin with this, we did an extremely simple experiment. We took the material that we used to solve the complex by EM, and we purified it in a 50,000-fold molar excess of cyclic AMP. And you can see that the catalytic subunit, although somewhat reduced, remained within the complex, suggesting that full occupancy of the cyclic AMP binding sites still retained this complex. We actually solved the structure of several Ecap pKa complexes using EM, and again using a technique that David mentioned earlier, we looked at the stoichiometry of these complexes using native mass spectrometry, which is a very quantitative way to look at the ensemble of complexes and the valency of cyclic nucleotide bound to the complex. And I'll draw your attention to these complexes here, where we have an intact pentameric complex, an intact pKa holoenzyme complex, with anywhere from 2 to 4 molecules of cyclic AMP. So this complex could be retained at some level when you looked at it in vitro. The next step was obviously to look inside cells, because it's a completely different milieu in situ. And here we're looking at an anchoring protein, which I'll talk a lot about, this molecule called Ecap 79, with the regulator and catalytic subunits. But now we used physiological agonists to activate the pathway, and you can see the pathway is activated here, where we're using a phospho-PK substrates antibody, and then monitor the composition of the holoenzyme complex when it was bound to an anchoring protein. And the important point was, even with a physiological agonist isopaterenol, which is a heart stimulant, you retained the integrity of the complex, although you got full signaling. The only way you could release the complex was when you created supraphysiological levels of the second messenger by adding a drug that blocks phosphodiesterases. And these are actually used clinically, sometimes in end-stage heart disease. They're pretty potent compounds. But again, Rolopram would do this, but interestingly, another isoform for a different phosphodiesterase had no effect. So this allowed us to conclude that cyclic AMP stimulation with beta agonists promotes minimal dissociation of the anchor pKa. So we really wanted to look at this inside a cell using a different technique. And this is where we took advantage of intermolecular FRET, where we developed these probes where basically you could look at fluorescence resonance energy transfer. If the molecules dissociated, you would lose the signal when you stimulated the cells with isopaternal. What you're looking at here are two cells that have been challenged with isopaternal, and there's little or no dissociation of the complex based on the FRET response. And you'll see this a little bit more clearly here when you look at the amalgamated data when we looked at many different cells. There's a minimal decrease, which is either movement of the cell or a very small decrease. The controls are to repeat the experiments using the phosphodiesterase inhibitor, Rolipram. You can dissociate the complex, but not when you use milrinone. We're also able to repeat this using a variety of different physiological agonists, particularly prostaglandins. And so it allowed us to conclude that there was a minimal release of the catalytic subunit in response to these two physiological agonists. So inside the cell, it was staying together. So the next question was, if this configuration is staying together, the regulatory subunits of the complex are believed to regulate the catalytic activity. But for this to be functional, you'd have to have some way for the kinase to be active, even if it's locked in this holoenzyme complex. So to do this, we took advantage of this sort of drug-inducible heterodimerization system, or rapalog technology as it's known, where you can add rapamycin or a less invasive analog that was developed by Ariad to pharmacologically lock two parts of a protein to hold the complex. So this is a pharmacologically locked complex within about 60 angstroms of each other. And then you can actually monitor kinase activity using an intermolecular FRET kinase activity reporter. And you can see in these two cells here, again, we're stimulating with isopaternal. We get a fairly robust response. We add rapamycin. There's no change in the response, suggesting that the kinase, when it's in the complex, is fully active, at least towards this particular substrate. So that allowed us to conclude that pharmacologically locking the subunits doesn't impede kinase dynamics. So one of the limitations of everything that I've shown you so far is we were looking at modified subunits, where we're looking at transfected cells, we're doing a variety of different things. So this is where we took advantage of proximity ligation to begin to look at the endogenous complex, where we used antibodies to the R and C subunits with secondary antibodies that are conjugated to an aptamer to create what are called PLA puncta, when you have protein-protein interactions that are somewhere between 200 to 400 angstroms apart, which was the kind of magic number here. So here's the control condition, stimulating with an agonist and stimulating with an agonist plus the phosphodiesterase inhibitor. And again, you can look at this as a maximum intensity projection, and you can see that you still have the complex even though you get full signaling within the cell. And it's only when you stimulate at supraphysiological levels that you lose this. And the reason I'm emphasizing that is that many of the studies that have been done up until now, and I mean there's somewhere in the order of 10,000 papers, including some of our own, that have used this condition, have been looking at kinase activity where it's non-physiological. The physiological system seems to be much more where you have a much more local signaling within this 200 to 400 angstrom range, which is really quite remarkable and surprising to us. So the sort of final part of this, where we wanted to really test this, was to take advantage of CRISPR-Cas technology, because there's a variety of ways that you can use this, but it's particularly amenable to taking out multiple genes in cells. So what we decided to do was to take out both regulatory subunits of the kinase. We actually did the type 1 and type 2. There are 4 genes there. I'm just showing 2 here. And a single copy of the catalytic subunit, the C-alpha gene. Now, the reason that we had to do that was that when we tried to do a quadruple knockout, we were never able to get any cells. We did this in several different cell backgrounds. And what we believe is happening is that this C-beta isoform, which represents approximately 10% of the kinase, was keeping the cells alive. So we were able to take advantage of this by knocking into this locus with a fused form of the kinase where the regulatory and the catalytic subunits were in one polypeptide chain. And you can see that here we have the triple knockout cells, we have the rescue with the quadruple knockout cells. You can see the expression of these. And the cell growth was restored when you had a form of the kinase that could not dissociate. It still responds to hormones. It still responds to the physiological agonists. So putting this together, what we really were beginning to think was the idea that if you have an intact kinase, the Ecaps become really, really important for directing the location of the kinase to these zones of 200 to 400 angstroms where the phosphorylation occurs. And we've called these regions Ecap signaling islands because they're islands within the cell where you can have this signaling to occur. So rather than a sort of diffuse effect, it's a highly localized effect. So that's really sort of drawn to the following conclusions. The anchor pKa has this very restricted range of action, somewhere between 2 to 400 angstroms. It could be slightly greater depending on the nature of the Ecap, because there could be intrinsic disorder within the anchoring proteins as well. Ecap pKa holoenzymes are active and intact inside cells. Very different than in vitro biochemistry, but again, it gives you much more regulation within the system. And localization by Ecaps is the key determinant for substrate specificity. These are broad specificity enzymes. They've got 2,000 to 3,000 substrates within a cell. If you're restricting their location, you can control their activity much more deftly. And that's the next generation for beginning to think about pharmacology, because we've got to target these domains to individually target different pools of the kinase. So the very final point in the last two slides are to deal with, if this is the normal situation, you'd begin to think that mislocalization of the kinase could have pathological consequences. And so very recently, we've begun to work on this notion, and particularly in the context of this endocrine disorder, Cushing syndrome, which is where the adrenal glands secrete excess cortisol. It can also be a side effect from excess taking of glucocorticoid drugs, where you have two forms. You can, in the pituitary, release ACTH, which then drives the release of cortisol, or there are ACTH-independent forms of Cushing syndrome, which are actually a consequence of mutations within the cyclic AMP signaling pathway. So this is where we really began to become quite interested in this, because these are point mutations that were identified by 4 different groups using precision medicine techniques that pinpointed the interface between the regulatory subunit in green and the catalytic subunit of pKa in blue. So a single point mutation or an insert, here's 2 examples here, prevent recruitment of the catalytic subunit to these Ecap signaling islands. And this is because you actually have steric and hindrance within this protein-protein interface. There is a strong mechanistic explanation for this. And again, we could begin to look at this biochemically by using CRISPR-Cas to introduce a single allele of this mutation into these cells, and it abolishes the ability to be recruited to the Ecap signaling complex. The cellular consequence of this is that these cells have a different subcellular distribution of the kinase activity. This is the normal kinase activity. It's excluded from the nucleus. But if you have a single allele of this point mutant, you see a much more regional distribution, suggesting that you've got mislocalization of free and active kinase that's kind of wreaking havoc within the cell. And in this case, it causes secretion of more cortisol from these adrenal cells. So finally, what we've been wanting to do is to try and look at this in real time. And so we've been taking advantage of tagging the catalytic subunits with a photoactivatable RFP so that we can stimulate for a pulse of light and then watch the movement of this. And then also using CFP, we've targeted the Ecap. So there's 2 examples here. And this is using structured illumination, where we're actually looking at a membrane-targeted anchoring protein, Ecap79 CFP. And you can see that the signal is fairly restricted. When we put the point mutation, this just shows the quantitation. The quantitation is kind of hokey. It's just measuring the distance from the plasma membrane. If we look at the point mutation, you can see a much more pronounced effect. There seems to be exclusion from the nucleus, but this could be because of the size of the catalytic subunit under these conditions. Just to look at this in slightly more resolution, these are single shots from these movies. You can see it's the same effect. And then finally, for good measure, we've done the same thing using a different anchoring protein. In this case, we've targeted to the mitochondria. Here's the wild type situation. And here's the mutant situation, where you see this distribution. So it would appear that mislocalization, at least in the context of Cushing syndrome, and not being retained within Ecap signaling islands can have pathological consequences. So with that, I will stop and give credit where credit is due. The vast majority of this work was really done by Donaldson Smith in my lab, and some of the more recent imaging is work that's been done by Mitch Omar. Thank you very much.
Thank you. We have time for some questions. Please raise your hand and we'll get a mic to you. Here's the first hand.
So that was really cool. Why is it selective to the adrenal gland to have the disordered A kinase signaling as opposed to many other cell types?
Well, I think it does happen in other cell types. We've just really begun to focus because these mutations only occur in the adrenal gland, which is interesting.
But presumably the mutations are germline, right?
So they're germline mutations, and they also happen in adenomas that you get in those adrenal cells. You can also get them in pituitary tumors as well, but that goes through the ACTH-dependent effect, which is an added endocrine element to this.
So is this, was it 79? I forgot which ACAP it was, but is that selectively expressed in the adrenal, or is it more widely expressed?
It's more widely expressed, but it's expressed at a fairly high level in those cells, as is the DPCAP1. So again, that gives you that added element of selectivity because you've got the differential expression of the ACAPs that can be targeted to different organelles and very precisely targeted to different organelles.
Thank you.